Hello everyone, welcome back to our channel. Get ready to dive into today's topic, which is CSIR Net Life Science Unit 5 Important Previous Year Question Papers with Solution. Today's topic is fertilization. But before we jump in, don't forget to hit that subscribe button and ring the notification bell so you never miss out on our latest videos. Let's get started. The first question from this topic is from CSIR Net December 2017 question paper, which is asking about the fertilization in sea urchin. So this is the diagram or the graph for the unfertilized egg where two components are there. One is the membrane potential. As we know, the resting membrane potential for a regular cell inside the cell membrane is minus 70 millivolt. This is what you can see here. And the component CBR1 receptor is high. Sodium ions inside the egg and potassium ions, they are kept low before fertilization takes place. The question is asking which one of the following graph best represents the condition within an egg 1 to 3 seconds after fertilization. Here two keywords are to be focused on. One is within the egg, which means they are asking inside the egg. Another keyword here is after fertilization. Also the timestamp plays a role, 1 to 3 seconds, which is where we know that the fast block to polyspermy takes place. Now, how to answer this question? You are supposed to know the key concepts for this. That is, whenever the sperm fertilizes an egg, there is a response taking place. The responses of egg to sperm are categorized into early responses and late responses based on the time limit in which they will take place. So, early responses, they are block to polyspermy, and late response is the activation of the egg metabolism. As all this while the egg was in a dormant condition, now that fertilization will occur, the egg will be activating the different processes, metabolic and also replication transcription processes. Block to polyspermy is of two types, fast block and the slow block. Fast block is an electrical change where the egg membrane undergoes depolarization. That means from minus 70 millivolt, its potential difference is going to increase to around 0 to 10 to 20 millivolt. So that will be positive, okay? That is why the polarization is lost. That's why it's called as depolarization. Slow block is through the cortical granule exocytosis. So how does the fast block to polyspermy occur? As I said, it occurs within few seconds. In the question, what it was given within 1 to 3 seconds. So yes, this is where fast block happens after the fusion of sperm with the egg. The sodium channels will open up in the egg plasma membrane. Sodium will flow into the egg causing depolarization, which will prevent any additional sperm from fusing to the plasma membrane. Obviously, this potential difference will not last for long because it has to form the fertilization envelope to support the zygote and the early embryonic development. So once the slow block appears or the cortical reaction happens, the sodium channels will close and other sodium ions and other positive ions will flow out of the cell and the plasma membrane will be restored back to normal minus 70 millivolt. So if you look at the graph from minus 70 millivolt, as soon as you add the sperms will be reaching towards the egg, and the moment the sperm enters the egg, it leads to a successful fertilization. And immediately you can see the change in the potential difference. Within 1 to 3 seconds after fertilization, the potential shifts in a positive direction. But it doesn't stay for continuously. After few seconds, you can see this is coming down. So once again, it will be restored to minus 70 millivolt. Since this is a block to polyspermy, if you prevent the depolarization, polyspermy will be occurring. So that's why here we can see that in this graph, the potential difference is increasing within 1 to 3 seconds and then again it is falling down. And regarding the sodium ion, since it is inside the cell, as we saw, sodium will flow inside the cell. That is why its concentration is going to increase within this time frame. Okay, but nothing happens to the potassium ion. That is why its concentration remains low. EBR1 is unchanged. Yeah, later the EBR1 receptors may be lost from the membrane as cortical reaction takes place, but not so fast as the one within the 1 to 3 seconds. That's why here the correct answer is option 3. Moving on to the next question. This is from CSIR Net November 2020 question. Several marine organisms release their gametes into the environment where sperm attraction and subsequent events lead to successful fertilization. Again, with reference to sea urchin, which one of the following statement is not true? So what you have to select over here is which is incorrect statement. Okay. For this type of questions, it is better that you apply the logic, whichever sentence you know for sure is correct. So elimination method is the best way to do like this. Let's start with the first one. Addition of Resact into a drop of seawater containing sperms. Right. So Resact is nothing but a chemoattractant. Okay. So if you add this into the seawater, it will specifically attract sperms of this particular species. Now, if you know about this chemoattractants, Resact is a chemoattractant in this species and Spiract is a chemoattractant in another species, S. purpuratus. Since these chemoattractants are species specific, hence you have to remember the names. That's why this is a correct statement. Second is IP3 is formed initially at the site of sperm entry. Okay. Now, first of all, how is IP3 formed and whether it releases calcium or not? To understand this, you must know the slow block to polyspermy. Now, slow block to polyspermy happens later after fast block within around 10 seconds to 1 minute of the fusion of the sperm to egg plasma membrane. Here, once the sperm will be fertilizing the egg, in the membrane there will be a receptor tyrosine kinase which will get activated. This receptor tyrosine kinase in turn is going to activate the phospholipase C enzyme. Phospholipase C makes a cut in the phospholipid PYP2. PYP2 means phosphatidylinositol 2-phosphate. It's a regular phosphatidylinositol which is a membrane phospholipid with 2 more phosphates attached with it. That's why it is phosphatidylinositol 2-phosphate. And phospholipase, after making a cut over here, it yields 2 components. One is the glycerol backbone with the 2 fatty acids still attached to the membrane. That's why fatty acid 2 is there, means diacyl connected with the backbone glycerol. So it is called as DAG. This is one component of the cleavage division by phospholipase C. And another component is inositol with its 3 phosphate groups. So 1 phosphate, 2 phosphate and 3 phosphate. So together these are known as IP3, okay, this molecule. Now IP3 has a binding to the calcium channels which are present on the endoplasmic reticulum, okay. These are ligand gated ion channels. So when IP3 binds with the ligand gated channels, the channels will open up and calcium flows out of the cell. This calcium is extremely necessary because this is what causes the cortical granules present under the plasma membrane in the cortex area of the cell where the cytoplasm is dense. These cortical granules contain hydrolytic enzymes. So when they release the contents outside into the perivitelline space, they will cause some changes in the envelope and also the perivitelline space, because of which other sperms will not be able to enter. Okay. So calcium is first released at site of sperm entry and it increases the concentration, not all at once, but slowly it increases as a wave. This calcium increases as a wave inside the cell. So here calcium causes fusion of cortical vesicles with egg plasma membrane, releasing their contents into the perivitelline space. And as we saw, inositol triphosphate causes release of calcium from intracellular stores. It is not coming from outside, it is released from internal stores in the cell itself, that is from the egg endoplasmic reticulum. So obviously, as we saw, IP3 is formed initially at site of sperm entry. As I said, it increases, the calcium increases as a wave and it will release the bonded calcium or the hidden calcium, the ones which were contained inside the endoplasmic reticulum. Now they are released. Calcium prevents docking of cortical granules. Yes, calcium doesn't prevent docking of cortical granule. In fact, it causes the exocytosis of the cortical granules. That means it will dock the granules to the cell membrane, which will help them to release the content. So since it is saying prevents, that is why this statement is not correct. Okay. Now PLC gamma was required to cleave PIP2 into IP3 and DAG. If you have inhibitors that are blocking this, then you will not be getting IP3 in that case. But if you separately microinject IP3 into the egg, then even if PLC gamma is not there, You need only IP3 to release the calcium and DAG to activate other processes. So even if PLC is not there, if you micro-inject the products that this enzyme was making, then there will be no adverse effect on the fertilization process. Okay, so that effect of inhibition can be overcome by micro-injecting IP3 into the egg. That is why even this statement is a correct statement. Moving on to the next question. So before that, let me tell you, if you want to study such differences, like just now we saw fast block and we saw the slow block, there are various points which you need to remember with respect to mammals, with respect to sea urchin. In case you want to have a ready-made book with all the points for quick revision, since your exam is coming, for quick revision you can refer the concept table book. As you can see, the points, all the points which are necessary, that's inositol triphosphate causes the release of calcium, calcium causes cortical granules. So like that we have for all the 13 units. This is unit 5, and even in mammals reference also, like which proteins are important and which all molecules will be preventing polyspermy. You can refer this particular book. You can contact our team for further details. Next question is from CSIR Net, December 2018. Here, human sperms are allowed to fertilize ova having non-functional ovastatin. So what is ovastatin? It is one of the components released from the cortical granules which will prevent the polyspermy. So following possibilities may be of significance in fusion of these gametes in case you have non-functional ovastatin. So which one of the combinations will represent the outcome of the above event? So since I told you ovastatin is released from the cortical granule, you must know the function of ovastatin. The function of ovastatin is to destroy ZP2. So it will be clipping of the ZP2. Clip of ZP2 means it will destroy ZP2. ZP2 is a component of the zona pellucida, which is the extracellular layer present outside the plasma membrane. This is in case of mammals. Zona pellucida has 3 parts. One is ZP3, ZP2 and ZP1. ZP3 is the main glycoprotein to which the sperm will be binding, but later it will also make contact with ZP2 to have successful fusion with the plasma membrane. That's why if ZP2 is not there, any more sperm will not be able to bind. So that means it will stop polyspermy. But the question is asking non-functional ovastatin. So if the ovastatin is not working properly, it will not destroy ZP2. So that's why C will be the cause. ZP2 will not be clipped by cortical granule protease, and because of which polyspermy will occur frequently. Okay, because ZP2, if it is still active, then intact, then many sperms will be able to fertilize the egg. However, the other regular fertilization events will not be affected at all, because for cortical granules to get released, at least one fertilization should occur, right? Then only the cortical granule exocytosis will take place. So that first fertilization, it will not be prevented and it will not be stopped by, it will not be affected by functional or non-functional ovastatin. So sperms will fertilize ova, sperms will bind and they will be fusing with ovum membrane, and CD9 protein will be able to interact with sperm membrane. So since CD9 is a fusogenic protein over here. Okay. So these are the cortical granule reactions which take place. There are some protein bridges which are present between the plasma membrane and the vitelline envelope. This is in sea urchin. When the cortical granules release the contents, the protein bridges are digested because one of the contents here is protease. And also there are some mucopolysaccharides which get released into the perivitelline space. Polysaccharides attract water, that is why a gap builds between the two membranes. The receptors become deactivated and a layer of hyaline deposits outside the membrane so as to make it a hardened membrane in order to support the further embryonic development. Okay. So these are the different cortical granule contents. Like protease, as I said, it will clip off the binding receptors and also it will dissolve the connections. Then mucopolysaccharides, they are sticky compounds, produces osmotic pressure. Peroxidase hardens fertilization envelope and hyaline forms a coating to support during the cleavage division. Ovastatin, as we saw, it clips off the ZP2. So as you can see, this is the membrane, mammalian egg membrane. We have the ZP1, ZP2 and ZP3. So when sperm binds to the end terminal of the ZP2 protein, okay, so these are the cortical granules which are present near the plasma membrane. When the cortical granules undergoes exocytosis, you can see that the ZP2 protein is now clipped over here, okay. So because of which any more sperm will not be able to fertilize. Then another question from 2016, Resat, Spiract are peptides released from egg jelly and they help in the sperm attraction. And you have to say which statement is not true over here. So as I said regarding the chemoattractants, yes, so chemoattraction, sperm attraction, action at a distance. So attraction of a sperm to the egg by soluble molecules released by the egg. So Resact is a 14 amino acid peptide in Arbacea punctulata and Strongylocentrosus purpuratus. In this sea urchin species, Spiract is the chemoattractant. Resact acts as a sperm activating peptide. When you have the chemoattractant released from the egg jelly layer, the sperm will sense this chemoattraction source and it will migrate to it. Okay, it will be flowing towards it. How does the sperm do it? It does it through a signaling process. So there will be receptors on the sperm for Resact or Spiract. So once they get activated, they can sense the chemoattractant in nanomolar range. The receptor, which is a guanylate cyclase, it's a GPCR kind of receptor which will activate guanylate cyclase. CGMP will be released and the fast CGMP increase causes transient calcium increase which leads to asymmetric beating and it changes the swimming direction. Then next, so the first statement is correct. Binding and acrosomal protein interacts in a species specific manner with eggs. Well, binding is a fusogenic protein. Okay, so we know that when the sperm will contact the jelly layer, it will undergo the acrosomal process, which is exocytosis. This will help digest the jelly layer and penetrate through it. Then the sperm will be extending the acrosomal process through the actin present beneath it. And there are some proteins which are present on the tip of this acrosomal process. Once it crosses the vitelline envelope, these fusogenic proteins present on the acrosomal process, this will help with the binding with the egg plasma membrane and these two membranes will then be fusing, releasing the contents of the male pronucleus inside the egg. So binding is the only sea urchin sperm protein known to mediate species selective sperm attachment to eggs. To completely different egg protein surface protein, as I mentioned, EBR1, they have affinity for binding and each one meets all the criteria to be a species selective sperm receptor. Okay, so whenever you have the acrosomal reaction taking place, you can see the acrosomal process is now bonded by binding. So yes, even this is a correct statement that binding and acrosomal process interacts in a species specific manner. A respiratory burst occurs during cross-linking of the fertilization envelope where a calcium-dependent increase in oxygen level is observed. Now, I told you about early responses and late responses. Early response was the block to polyspermy and late responses are events where sea urchin egg will be activated. One of the events that happen over here is increased respiration. So as you can see, the increase in oxygen consumption taking place. Oxygen consumption, if it increases, means oxygen concentration will be decreased, isn't it? If aerobic respiration is happening, oxygen is getting used up and that is where we can say oxygen concentration will decrease. But since in this question it is saying that oxygen concentration increases, that is why this increase word is wrong. Yes, respiratory burst takes place during cross-linking, this is correct. Calcium dependent, this is also correct, but increase is wrong. Now, in original question answer key, not the final, the original, the previous one, it was marked as only C as the right option. Okay, so this is the only statement which is not correct. But later they had dropped this question because on challenging the question, this option 4 also seemed to be wrong. But since there is no option as C and D in the options, that is why they dropped this entire question. Okay, so the correct answer according to the final answer key should have been C and D. Okay, Why? Because IP3, which is formed at the site of sperm entry, it will release the calcium leading to cortical granule exocytosis. Since it is saying it sequesters, no, it doesn't sequester. Sequesters means it is binding or it is hiding or enclosing. No, IP3 will release the sequestered calcium. If it said releases sequestered calcium, then it is correct. But since it is saying sequesters calcium, that is why even this statement is wrong. So observe carefully in questions. Each of the keywords are very important and that they will lead you to getting the correct answer. That's all for now. Thank you everyone for watching.